Hi everyone, I'm Jan from Chalmers, and I'll present my recent computational study about finding origin of transfer regions in plasmids. So a lot of plasmids carry transfer regions, but not necessarily any of the other conjugative protein components. It was recently shown, though, that such plasmids can still transfer with co-resident plasmids, so it's important to find these regions as they can point to the true plasmid mobility in nature. However, there are a few such methods, and recently we found a nice way to type plasmid mobility groups, defined by the relaxase enzymes, where the DNA structural properties of transfer regions are more conserved than the nucleotide sequences. So here we develop a new method for finding and typing transfer regions, an alignment algorithm based on encoding DNA into structural representations, which enable computation of the structural distance used for alignment and thus finding significant origin of transfer hits in target plasmids. The method is in fact more accurate for typing transfer regions than existing sequence-based methods. So we then type the transfer regions in a dataset of natural plasmids, finding thousands of new regions and uncovering an almost twofold higher putative plasmid mobility across twofold more whole species than is currently known. A very interesting finding is that well over half of these mobile plasmids carry multiple transfer regions from different mobility groups, so they can be mobilized by different conjugation systems, potentially linking multiple host ranges that are currently understood to be confined. In fact, we find that the amount of resistance genes and transfer regions carried by a plasmid are actually significantly correlated. So then a final analysis was to build and explore a potential cross-habitat transfer network based on these multi-ority plasmids, which shows it might facilitate the transfer of resistance genes from environmental reservoirs to human bacteria, and suggests that the observed transfer regions might be an important point of focus for future study and prevention measures. Thank you for your attention.